So today's webinar is jointly sponsored by the SREF Forest Health and Invasive Species Program and Emerald Ash Borer University. We are joined by Dr. Dan Herms from The Ohio State University. Dr. Herms will discuss the biology, ecology, and management of the emerald ash borer, or EAB, in natural and urban southeastern forests. This webinar is designed to help woodland owners, foresters, researchers, arborists, and concerned citizens learn how to identify EAB and discuss potential management options. The title of Dr. Herms' webinar is Emerald Ash Borer Biology, Ecology, and Management, and Implications for Natural and Urban Forest Areas in the Southeast. With that, I'll turn it over to Dr. Herms.
Thank you, Dave. Excuse me, and thank you, everybody, for joining us this afternoon. Emerald ash borer, of course, I'm sure that you're aware, is an invasive species that is fairly rapidly invading North America. It's in the genus Agrilus, which are the metallic wood-boring beetles. I think you can see where they get that name. And it turns out that the ecology and biology of emerald ash borer, the life history, is very similar to some native Agrilus species that you may be familiar with. For example, bronze birch borer and two-line chestnut borer. The larvae are referred to as flat-headed borers because of their very flattened shape. So as emerald ash borer continues to spread, it's killing millions and millions of trees. People ask how many trees has EAB killed, and I don't think anybody has any idea. There was an estimate completed in around 2007 that estimated between 50 and 100 million trees, and that number's increased exponentially, but it's killing trees in all environments. Healthy trees, stressed trees, there's golf course trees you can see here in irrigated environments, woodlot, street trees. The impact is comprehensive. So today I'm going to talk about the biology of emerald ash borer, its current distribution as we know it, diagnostics, how to diagnose emerald ash borer, some research we've done on the forest impact and ecology of the insect, also our research on host resistance and insecticides, and then I'll talk a little bit about management in urban and natural forests. So the host impact is very similar to its native relatives in the family Buprestidae in the genus Agrilus. They feed on the phloem and scar the xylem. You can see the kind of damage here, feeding just under the bark on the inner bark. All ash species that it's encountered so far in North America are susceptible. White fringe tree is also known to be a host in North America. There's a number of western species that have not yet been surveyed or encountered by EAB, and we don't know whether those are susceptible or not, but the western species that it has encountered, and I'll talk about this a little bit, are susceptible. So this feeding disrupts phloem transport. Ash has ring porous xylem anatomy, so the functional xylem is quite thin. So this scarring of the xylem also has an impact on water translocation, and as a result, ash trees die more rapidly in response to EAB colonization than species like birch, for example, do when they're colonized by bronze birch borer, which birch has a diffuse porous xylem anatomy, which consists of more annual growth rings. So EAB attacks trees of all sizes, anything big enough to physically house a larvae, half-inch diameter saplings to the largest mature trees. The life cycle takes 1 to 2 years in the Midwest to complete a generation. And the typical life cycle is a 1-year life cycle. I'll talk about that. And then I'll talk about the 2-year life cycle because that has really important implications for colonization of more southerly environments. So adults in Ohio begin emerging in late May and into June and July. The peak is in early June. Emergence begins about Memorial Day. The adults feed for a couple weeks on foliage, 10 days to 2 weeks. They feed on foliage, and they have to do that and call it maturation feeding before they're reproductively competent, before they can mate and lay eggs. So after they nibble on the foliage for a couple weeks, they'll lay eggs on the bark or in bark crevices. The larvae hatch and then chew their way through the bark into the phloem. And the larvae feed throughout the summer they grow slow, they can overwinter, but in this area, most larvae overwinter as pre-pupae. They complete their larval feeding, overwinter as pre-pupae. They're done feeding. They create these chambers. Here you see pre-pupal larvae in a cross-section of a piece of bark. So they'll pupate. They'll construct pupal cells in the outer bark or shallow inside the xylem, no more than a ½ inch into the tree. They pupate in the spring, and then adults emerge. So that's the 1-year life cycle. Now, if larvae are growing slowly or the eggs were laid late and they don't reach the pre-pupal stage by the winter, then they can overwinter in any larval stage, second, third, early fourth instars. There's 4 larval instars. The pre-pupa is a mature fourth instar. So if they don't reach the pre-pupal stage by winter, they'll overwinter and as larvae complete their development the next spring. And so this kind of shows, it looks a little bit complicated, but the 1-year life cycle, or this outer ring, the 2-year life cycle. So each half of this circle is 1 year, going from summer, adult emergence, autumn, winter, spring, summer, autumn, winter, spring. We just talked about the 1-year life cycle. But in healthy trees, trees that can still mount some sort of wound response, callus tissue, so forth, larvae will grow slow, or in cold climates, larvae will grow slow. And if they don't reach the prepupal stage, then they'll overwinter as larvae, finish their development in the spring, and then become prepupae. But they will not emerge until the prepupa has experienced wintertime, right here. So the prepupa apparently has to experience cold weather. Now, this has been demonstrated for bronze birch borer, and it seems to be the case for emerald ash borer as well. And so what that does is synchronize the 1st year and the 2nd year life cycle, so they both pupate in the spring, and adults emerge at the same time, which facilitates mating and resynchronizes the life cycle. So this has implications for the southern environment, because one question is there just a distribution that's too far south, where there's not enough chilling for the prepupae to break diapause and pupate? Or can EAB evolve a non-diaposing strain, in which case the population could become asynchronous, or maybe 2 generations a year, and populations may grow much faster? This would be an interesting question for researchers in the South to address. This could constrain EAB, or it could facilitate more rapid population growth, depending on how EAB responds to these warmer climates where the winter doesn't have the chilling. The economic impact of EAB has already been substantial. It's already the most expensive invasive forest insect ever introduced to North America, and most of that impact has been the cost of removing and disposing and replacing dead trees in urban environments. That's really where the big impact has been in Michigan, Ohio, Indiana, Illinois. And if you look at a continental scale, the EAB invasion is actually quite young. So the economic impact is going to continue to get extremely devastating as EAB continues to spread. EAB is native to East Asia, Japan, Korea, Siberia, China. It was introduced almost surely from China based on molecular genetics. There's also an invasion occurring in Russia. It was detected in Moscow in 2004, and it's spreading through Europe as well. So this is a second invasion, one in North America, one in Eastern Europe. In North America, outlier infestations such as those in Georgia, Colorado, I'll show a distribution map here shortly, have been the result of movement infested ash trees. EAB spreads much slower in a natural. The natural spread is much slower. It is a strong flyer, and it is capable of long flights, but typically EAB infestations don't spread very quickly on their own, but they are spread by nursery stock firewood and so forth, and quarantines are established to try to limit that. The blue lines indicate the borders of the quarantines to reduce that natural spread. So here's the known distribution of EAB, and I emphasize known. This map changes quite frequently. Colorado is the most western remote infestation here in Georgia, Louisiana, Arkansas, the southernmost infestations. It was established here in Detroit. It was discovered in 2002, but it was been there probably at least since the 1980s, and we found trees that died as early as 1994, remote from the point of introduction. So it's been there a while, and it's currently known to exist in 26 states. You can see the distribution here. Overlaid on the green is the distribution of ash, and it extends well into the south, as you know, up into Canada. This doesn't show the Canadian distribution of EAB, but it's in Quebec and Ontario as well. So we're looking at a continental scale invasion, and here are the western ash species. So symptoms identifying EAB. One are the thinning canopy. Note that it's not dieback from the top down. The trees still have foliage at the top of the tree, but the canopy progressively gets thinner. This is pretty characteristic. There, you do see dieback, but that's not the typical pattern. And I'm, after looking and rating trees for more than 10 years, think that that's probably when you see dieback, it's related to another issue that's going on simultaneously with the tree. So you get this classic canopy thinning. I'll show some more pictures of that. Epicormic branching from the trunk, very characteristic when the tree is healthy and it's first colonized, and then this girdling response has the tree push out these sprouts. And then when a healthy tree dies, you get this vigorous sprouting from the roots. You don't see this as much in the forest. You see this in open-grown trees, but we have not seen this in the forest. In the forest, we see little wimpy sprouts that pretty quickly die. But in open environments, if the tree dies quickly, there's enough vigor in the root system that these sprouts can grow and themselves become big enough to be infested. Of course, D-shaped exit holes, very characteristic D-shaped exit holes. The galleries just under the bark filled with frass. And then the tapeworm-like larvae with these bell-shaped segments. So these are all very distinctive in ash. There's no native borers that do this kind of thing in ash, and there's no native larvae that is flattened quite like this, nor is there anything that produces these very distinctive D-shaped exit holes. Now there are native borers, and these are extremely common, and as we found as EAB spread in Ohio, when people find a dead ash tree, they always find borers in it. Often these are native borers. So the clearwing borer, the banded ash clearwing borer, the larvae feed in the sapwood and heartwood. They expel frass at the base of the tree, which EAB doesn't do. They produce round, larger exit holes. Round-headed borers you find in dead and dying ash trees. This is the red-headed ash borer. There's other species. They're fat and plump, not flattened. They also bore into the heartwood, which EAB doesn't do. The exit holes are distinctive. So here's the D-shaped exit hole of EAB. The clearwing borer holes are round and they're larger. The round-headed borers, like the banded ash or the red-headed ash borer, are larger and oval. And furthermore, they penetrate into the tree. You can stick a thin, flexible wire into the tree through this hole, whereas EAB exit holes truncate right in the bark. Or at most just inside the phloem, so you cannot penetrate those holes with a thin wire. Unusual woodpecker activity is a good early warning sign. So woodpeckers, downies, red bellies, red-headed, hairy woodpeckers feed on prepupae over winter. The overwintering stage is when, in Ohio anyway, when most of the woodpecker predation occurs. They're feeding on the big plump mature larvae. Here's severe woodpecker damage, and these are all woodpecker predation. They're not knocking bark off at random. Here is some light flaking of the bark. And if you see this in the canopy of an ash tree, you should be concerned. At least in Ohio, there is no other insect in ash, a healthy ash tree, that woodpeckers are feeding on. If you see this, the tree's infested with EAB, almost certainly, in Ohio. Other early warning signs, bark splitting. Here you can see the bark starting to split. You can see a gallery under there, these little bark splits. So this is a result of when a vigorous tree is attacked. It produces this callus tissue over the bark. You may be familiar with this with bronze birch borer and other, thin bark trees. Here the bark is too thick to see the ridges, but that callus formation pops that bark. The bark's not flexible, causes these splits to form. And this will happen while the canopy is still healthy. You can look up in say the 3 to 4 inch branches and see some of these cracks. Starting the canopy still looks healthy, and you can have really good luck implementing insecticide treatments at that point. The trees can tolerate a fair amount of feeding, not 50 larvae per meter squared, but you can detect an infestation in time to start treating it. If you know what you're looking for and you look closely, you can detect an infestation in time to start treating it before the tree begins to decline. So here's the distribution of the major ash species in eastern North America. And you can see green ash extending down into Florida, white ash penetrating the southern United States. Blue ash is more regionally endemic, and black ash extends fairly north into Canada. So as EAB continues to spread in the east, it's going to be a continental scale problem. And so, we've been interested in ecological impacts of this widespread, and we'll see simultaneous ash mortality. And where ash occurs, it tends to be very common. I know in Ohio, ashes represents about 10% of the stems in Ohio's forests. So we've been interested, what happens when 10% of the trees die. And so we've looked at initially the susceptibility of forests. Are all forests equally susceptible to invasion? Would EAB not invade a low-density ash forest? How fast do the trees die? Successional patterns, regeneration, effects on native flora and fauna, including invasive plants, are questions that we've been looking at. And we started our research here right at ground zero. EAB became established initially here just on the west side of Detroit, in a western suburb. We worked in this watershed, Huron River watershed. This is heavily forested area. We established 38 forested stands, 3 plots per stand. So these forests are have been impacted for the longest of any forests in North America, which is why we were interested in working there. And we worked across a moisture gradient from upland white ash forest to riparian green ash forest to black ash swamps that occur in standing water. And what we found is that the ash trees died quite rapidly. When we started 2005, 40% of the ash trees were dead. These are trees greater than 1 inch stem diameter, so anything bigger than this. 40% dead. By 2009, 99.7% dead. You see this little drop. This is these saplings entering this 1 inch that were too small to be infested, and they're growing and entering the 1 inch diameter category. So you can find this paper online that documents these patterns of mortality, regeneration. And another thing that we did was dendrochronology. We went through and found big dead trees in our plots and dated them through the 90s. The oldest one we found was killed in 1994. So this suggests the EAB invasion has been like other biological invasions. Starts out with a low lag phase where densities are low, and then it reaches an inflection point and increases exponentially. So the solid line we measured, the dotted line we infer based on knowing that trees began dying at least as early as 1994 in this plot. And so you can see the trees die very rapidly. This is that simultaneous mortality. most of the trees died in a 3-year period. Over 60% of the trees died, 70% of the trees died in just a 3-year period. So it starts out slow, but then when it picks up steam, it goes really fast. And the same patterns have been observed in urban forests, which has resulted in some municipalities just being overwhelmed by too many dead trees for them to handle. So as you might guess, when you have 99.7% mortality, there was no relationship with ash density. And we worked across a gradient of very low density to very high density, and just no pattern. All the trees died. So ash doesn't seem to be able to hide at low density. In fact, there was no relationship between ash mortality and any of these community level variables, which also suggests that there's limited potential to prevent ash mortality via silvicultural practices. We've looked at ash regeneration. What is the future of ash in these plots where 99.7% of the trees have died? So we've looked at seed bank dynamics, seedling and sapling demography, and EAB populations. Do they persist even after the mature trees have died? We found there is no ash seed bank. No seeds persist in the soil. They're fleshy. They either germinate or they disappear in the year that they fall. So here this just shows this number of viable seeds per hectare. After 2007 into 2008, we found no seedlings, and since then we've also found no germinating seedlings. No seeds in the soil, no germinating seedlings with cotyledons. Not a one. And every year we have our summer workers, we put out a bounty. If they can find a new seedling, they get a prize, but nobody has claimed a prize yet. So there are these established seedlings. These were seedlings that were present prior to the EAB killing their parents, and saplings, but no mature trees and no seedlings. So we definitely have an unstable demographic pattern. The whole future is in what we call in this paper, this orphaned cohort of established seedlings and saplings. And so what will be the fate of the orphan cohort and ash in these sites? And so these are all ash seedlings, a few other things sprinkled in, sassafras. But the vast majority, almost all of these are ash seedlings, this carpet of ash seedlings that were established before EAB killed the parent trees. And so they're persisting. They're too small to be colonized. EAB is still persisting. We find very low levels on these purple traps that are used to monitor for EAB. So the population has dropped dramatically, as you might expect, with the carrying capacity declining as all the larger trees in the Phloem area has dramatically decreased. And so, will EAB be able to persist on these saplings? As these seedlings get big enough to reach that 1/2 inch to 1 inch diameter, they can support a couple EAB females, and then the tree will die. Will any sapling survive to reproduce? And will EAB become locally extinct because of the lack of resource? So those are questions that we're interested in. And a wild card in all of this is the biocontrol agents that USDA, APHIS, and U.S. Forest Service has released in many locations, including in Michigan. And these parasitoids, part of this classical biocontrol program, are not having any detectable impact on slowing the population, the spread of the population, causing outbreaks to crash. But it's a very different question as to whether they can regulate very low density EAB populations after they crash on their own because they've fully exploited their resource and killed the ash trees. And in Asia, EAB exists at really low densities naturally, and so these parasitoids have evolved under low density situations. So that'll be interesting to see, if a dynamic equilibrium establishes between the orphan cohort, EAB, and these parasitoids. So the conclusions from this point are that there's comprehensive mortality, there's no regeneration, just this orphan cohort, and we don't know how the dynamic is going to play out yet, but we're continuing to look at that. We've also been interested then in the effects of this widespread simultaneous formation and gap formation on native plants and animals in these forests. What are the ecological impacts of all this ash mortality? And Kamal Gandhi, who's a forest entomologist at the Warren L School at the University of Georgia, was a postdoc with me, and she published this review paper hypothesizing and documenting ecological impacts that other invasive species have had. And as trees die, you can imagine all kinds of interacting effects that cascade down to affect successional trajectories in native flora and fauna. An example, again, is this banded ash clearwing borer. This native borer requires trees at least 4 inches in diameter, and our pheromone trapping suggests that it's gone locally extinct in those forests of southeast Michigan. And in fact, we've documented in a literature review, Kamal and I, that there's at least 44 species that use only ash, so they're also at high risk of extirpation. And there's a number of other species, at least 286 we documented in the literature, that use ash to make their living. So you can also imagine indirect impacts, and Kamal looked at ground beetle communities in response to gap formation in these forests. Ground beetles are considered very sensitive environmental indicators of environmental change, and she found indeed that their populations were reduced in these gaps compared to the closed canopy forests, so documenting another indirect effect. We've also looked at invasive plants with Kathleen Knight, the U.S. Forest Service, colleagues John Cardina at Ohio State, Wendy Klooster, PhD student, and this concept of invasional meltdown, which is where one invasive species facilitates the success of another invasive species, in this case invasive plants. And in Ohio, these tend to be woody shrubs that are limited by light, so that you find them on the edge, but not so much deep in the forest. But if you start having these gaps form simultaneously, these plants tend to be bird distributed. Could they hopscotch their way through the forest? And so we're looking at that. And we also found that when they are in the forest, once they get in a gap, their growth rate doubles, whereas the growth rate of the native plants, which are probably adapted to shade, which is why they're there, is not as responsive. So the invasive plants are favored by the gap formation. So in Asia, EAB is not a big pest. It's a secondary pest that colonizes stressed trees, much like bronze birch borer, two-line chestnut borer in North America. And so we started a research program immediately, working with Eric Rebeck and Dave Smitley at Michigan State to identify resistant germplasm, to test the hypothesis that Asian ash trees are resistant by virtue of their coevolutionary history with EAB. And we're also looking for native lingering ash, those rare native trees that didn't die. Are they resistant or lucky? Working on mechanisms of resistance, identifying genes, and all of this to inform a breeding program to develop resistant ash that can be used in restoration of native and urban forests. And our first finding was that Manchurian ash, which has a coevolutionary history with EAB, is indeed resistant, much more resistant than North American trees that you can see here in this common garden study in Michigan. We've expanded the study to look at a number of North American species: white, green, this is Oregon ash, Fraxinus latifolia, West Coast species, green ash, blue ash, Manchurian ash, this hybrid that's in the nursery industry, a black ash times Manchurian ash hybrid, and then these European species, which are of interest in Europe because of the expanding EAB population there. And what we found is that Manchurian ash and the hybrid, Manchurian ash times black ash hybrid, and blue ash are much more resistant than the other species. Blue ash is the most resistant of the North American species that have been evaluated so far. It's not as resistant as the Asian ash. What this shows is mortality. Blue ash are continuing to decline, whereas the Manchurian ash aren't. It was initial mortality, Manchurian ash related, I think, probably to transplant stress and some deer rubs and so forth. But blue ash is continuing to decline, but at a slower rate than American ash, and then these other species, highly susceptible, 100% mortality of the European and Oregon ash, so the West Coast is highly vulnerable as well. We've been interested in the phloem chemistry. EAB feeds in the phloem, and so the chemical defenses, this is Don Cipollini at Wright State University. We've documented that Manchurian ash has a very distinct phenolic profile in the phloem compared to white ash and green ash, and we've worked on trying to identify, what are the mechanisms that make resistant Manchurian ash to EAB. And we've identified a number of traits that are unique to Manchurian ash, overexpression of proteins, and in particular, some exciting results that we've found very recently. Chad Rigsby, a student working with Don Cipollini at Wright State University, is that the phenolic compounds are oxidized much rapid and a more intense rate in Manchurian ash and that black ash, and this causes intense stress to the midguts of larvae. And so we think we're starting to hone in on the mechanism, and we've been able to confirm this with using artificial diets as well. If you're really interested in the research that we've done on resistance, we've reviewed the papers that we've published in this review paper, and goes into great detail on what we've learned about mechanisms of ash resistance to EAB. Lingering ash, so I mentioned 99.7% of the ash trees died in southeast Michigan forests, but here's one that didn't. So is this tree naturally resistant? Was it lucky? Is it going to die? We don't know. But when we find those trees, we map them, and the Forest Service has collected scion material, propagated them so they can grow them up large enough to evaluate their resistance under controlled conditions. So we'll see if this is if these lingering ash are a source of resistance genes in the native populations, which would be kind of ideal. So, to add to the conclusions, Manchurian ash and probably other Asian species which we're evaluating now are source of resistance genes. Maybe these lingering ash are too, and this breeding program is underway in early stages. I've also looked at insecticides to control EAB in urban forests, street trees, shade trees, so forth. And we've looked at systemic insecticides, which can be applied as soil treatments, trunk injections, or these systemic basal trunk sprays. And I'll talk about these studies. We've been interested in these questions. Will systemic insecticides work on larger trees? Most of the early work were done on small trees. We're now working on trees larger than 50 inches in trunk diameter. Rates, timing, duration of efficacy, and so forth. So we use this in these multi-year studies where you can't cut down trees and count larvae. We use these canopy decline rating scale. Dave Smitley published, and all the researchers use the same scale so that we can compare our results directly. But again, remember I talked about the diagnostics and the thinning canopy. So here, this is what this canopy thinning looks like, as opposed to top-down dieback that you might think about with, say, bronze birch borer. You see, this progressive thinning, more light, smaller leaves, fewer leaves, more light penetrating the canopy. There's still foliage at the top. So this is something you want to start developing a search image for. And once you do, boy, you can really pick out these ash trees in areas where you can pick out the declining trees amongst the healthy trees as EAB begins to invade. So we studied emamectin benzoate. It's a trunk-injected insecticide. There's different formulations out there now. At this time, there was only one, Triage. It's off patent now, and there's several formulations, some restricted use, some not. And we looked at different rates. If you're familiar with the rates, they go up from this low up to high, medium high, and even higher, 15 mils per inch, but we found it's hard to get that into the tree even. and our studies show you don't have to go that high. We treated the trees once, 2006, and then just monitored them to see how long they were protected. And what we found is that very solid 2 years of control, and even 3 in some situations. Other studies have also found 3 years of control. These are pretty large trees, 20 to 25 inch, under high pressure. So the white line is the untreated control, experimental control, not insecticide control, but the check. And what we see in 2006, none of the trees showed any decline. 2007, after the first year, this is 1 year of control, and you can see that the untreated trees are starting to show some decline, but not much. But in the second year, dramatic, rapid decline. In the third year, they're basically all dead. So you can see the trees go from less than 10% canopy decline to 95% decline in just 2 years. So this means, that you really want to start treating while the trees are still healthy. And you can see that all the triage treatments provided good control after, through 2 years. But into the third year, these started to decline. And we recommend that under high pressure, when trees are dying all around, that you treat every other year. Under low pressure, you can get away, I think, with treating every 3 years. This is our recommendation anyway in Ohio, using triage. So here's our study site. You can see kind of what we're dealing with. This is when we started. The trees are looking healthy. 2009, here you can see untreated trees, or treated trees looking good, but the untreated trees declining rapidly. See the dramatic canopy thinning, rapid mortality of the trees. So we started the treatments just in time to preserve the healthy trees. We didn't have to treat for years ahead of time. We've also looked at imidacloprid soil drenches, and we apply it just by mixing in a bucket, pouring it right at the base of the tree where you get better uptake than going farther out to the drip line. Here's our study site. We looked at 2 different rates. This is the maximum rate for most insect pests, and then there's a 2x rate, was developed based on this study, for certain hard-to-control insect pests on trees greater than 15 inches in diameter. So I'm going to call these the 1x rate and the 2x rate. And we looked at spring and fall treatment. Arborists like to treat in the fall because they're busy in the spring. Fall doesn't work as well, as we'll see. And then spring treatments. And so here's what we found. Again, we treated these trees every year. Imidacloprid only provides 1 year of control. We treated every year through, actually now 2014. No, 2013 was the last year we treated. And so they were all healthy. In 2007, showing a little bit of decline. And then the untreated trees declined rapidly. The trees treated with the low rate in the fall also declined. Then they were removed as hazards. But the other trees are looking, came through the outbreak and look really, really good. 1x in the spring, 2x in the spring, 2x in the fall. So if you treat in the fall, all things being equal, you have to use a higher rate on trees of this size. These are the biggest trees that I know of that have been tested with imidacloprid. But this suggests that since the 1x worked as well as the 2x, that you can go bigger on the 2x rate, which you're allowed to use on trees greater than 15 inches DBH. So here's our study site. 2006, the trees look great. 2009, you can see trees that were treated and trees that are not treated. 2013, the trees that were not treated have all been removed. So it leaves the street with these healthy trees. And all the untreated trees in Toledo are dead. These are the biggest healthy trees left in the city. EAB population has crashed. And we've now stopped treating those trees and are just monitoring. We transitioned first to treating half the trees every other year, but we're just going to stop treating now and just monitor. And then start treating when we start to see the earliest symptoms. EAB population is so small that they won't overwhelm these trees. So we want to see, how often we have to treat these trees. Maybe not very often. Maybe they can be preserved very cheaply now. So that's what we're looking at here for the long term. We've also tested Safari Dinotefuran, basal bark spray. You treat the tree. It's a neonicotinoid. I should mention that the soil, I'm going to back up, these soil treatments, we always applied right after ash trees flower. So ash is wind pollinated. It doesn't produce nectar, but the male trees produce pollen, and bees will collect it, although we've never actually seen a bee collect them. There are reports in the literature, but we treat it after they flower, which right before the leaves come out, and been very successful with that. So I think it's a bee-friendly approach as well. And so dinotefuran, the basal systemic spray, it penetrates the bark and then is translocated up through the canopy, and then soil treatments. And what we found is they work equally well. There's a dose effect. The high rate is better than the low rate, but the soil treatment and the basal trunk spray at the high rate are both very effective. We've tested really large trees. Now these are even bigger than 50 inches. Here's a treated tree in the neighbor's yard. These people, not part of our study, didn't treat their trees. We work with the arborist, but here's a treated ash tree. Here's a treated ash tree, and this is near Cleveland where ash mortality is really high. And here's percent canopy decline of these trees treated over every other year with emamectin benzoate. So even this low rate is, you don't have to use the high rate. The 5 mil rate, very effective on the biggest trees that we can find in the landscape. So as far as managing EAB infestations, the options are fairly limited, and can do nothing and let nature take its course, which in forest, that's often the case. You can't really do that in the urban environment. Remove and replace. Tree inventories are really important for that. Sustained insecticide treatments, as we were just talking about, and increasingly municipalities are integrating all of these. In wild areas, away from trails where their hazards aren't a concern, the trees just die and they fall down. And they fall quickly. Ash trees get very brittle. They don't produce standing snags for very long. Within a couple years, they start falling. So removal and replacement in the urban environment, you want to do this. If you know you're going to replace the trees, it's much safer to do it while they're still alive. After they die, they become very dangerous to climb and remove, and they shatter when they fall. The insecticide treatments are very effective. Quarantines prevent the unnatural human-assisted spread, which I think has reduced dramatically from the late 90s and early 2000s when we didn't know about the risk. I think the quarantines have really slowed the unnatural spread, but natural spread's going to continue, and so is unnatural spread. We showed this exponential increase in ash mortality. This has implications for urban forests because records from places like Fort Wayne and so forth show the same thing happens there. So if you do nothing, we know what's going to happen. At first, very little. But then you're going to reach an inflection point, and most of the trees are going to die in a very short period of time. This has happened over and over again. Now, the insecticides work very effectively, and they can be used as a tool in urban to manage a population of ash so that you can bend this curve to your will. You can stabilize mortality at a certain point if you want to maintain half the trees. You can delay mortality, remove trees according to your budget and timetable. You can prevent mortality altogether. You can make this curve look like anything that you want it to look like. You can model the population. You can remove undesirable trees, trees that are not so healthy, and preserve healthy trees. You can preserve healthy trees for a while. You can, if you preserve them too long, you may be stuck with them because the EAB population will crash, which can be a good thing. So, there's a lot that you can do, and there are tools now where you can, model these responses, and I'll talk about that in a second. But first I'll point out that some municipalities are choosing to treat the largest trees and remove smaller ones. The smaller ones are easier to remove and to replace their ecosystem services in a short period of time. If you look at the demographics of street trees in an urban forest, the chances that you're going to remove these trees and then replace it with a similar forest over time is exceedingly low. Street tree survival rates are just not good. And so if you had this, my question is, why would you want to get rid of it and bank on possibly, the probability that you can regenerate this? The probability that a tree reaches a large size in an urban forest is low, and this has been documented in demographic studies. And the economic value of these trees is exponentially greater than smaller trees. And so you can model these things. You can use this Purdue EAB cost calculator that Cliff Sadoff has developed, this website, and you can model the cost of treatment versus replacement. You can put in your own variables, your own cost variables, pesticide costs, labor costs, so forth. Generate the ecosystem, the value of the ecosystem services, or not include that if you want. But these public economic analyses, and there's a couple more now, show that it's actually more economical to treat large trees than to remove them. And we've been looking at management in the forest situation. We're working with the Five Rivers Metro Parks area in Dayton, where they're treating 600 mature ash trees in the forest. These are reproductive trees, males and females, to see if they can conserve ash regeneration and genetic diversity. This is Aaron O'Brien, a graduate student of mine, is looking at the effect of these treatments. And can you continue to produce seed production and new generation, and and maintain some sort of an aftermath forest, maybe in conjunction with the parasitoids. So it's kind of like what they're doing in Great Smoky Mountain National Park with hemlock woolly adelgid and treating hundreds of thousands of hemlock trees with the imidacloprid to preserve that resource. So a lot of what we've talked about that I just talked about, the insecticides, is available in this bulletin. Dave mentioned emeraldashborer.info. It's a great source of EAB resources, including this insecticide bulletin. We now have a second edition that we published last year, which is updated. We also have this bulletin available, peer-reviewed, highly peer-reviewed bulletin that discusses potential ecological side effects of the insecticide treatments, in case you get these questions from clients or you have the questions yourself. So with that, I've pretty much been over these conclusions, but I would be happy to entertain any questions that anybody might have. So if you're still out there, I appreciate your attention, and don't hesitate to email me if you have a one-off or a one-on-one question that you want to talk about later.
All right, thanks, Dan. Great webinar. We've got a few questions that have come in. Everyone that's still here, if you'd like to submit your question in the chat window, I will take that and I will moderate. But let's start with some of the questions we've already got. Dan, the first question is this. After the first destructive wave of ash mortality, and you have a few ash left and low numbers of EAB in the area, what type of population dynamics come into play then in the absence of new introductions or biocontrol?
That is a really good question, and I don't know the answer to that, but we're looking at it. We've tagged and we're monitoring a large number of individual saplings that were too small initially to be colonized to see if, and we've sampled EAB with traps in these sites. So I don't know if EAB will form some sort of dynamic equilibrium with the saplings, or a large tree can produce thousands of EAB adults. A sapling can produce 2 or 3, and will EAB ultimately go extinct? And then if so, will EAB kind of reinvade? So good questions. It hasn't been here long enough for us to know the answer.
All right, the next question, Dan, is how well do pre-adult EAB survive tub grinding or chipping? So research has shown that if you chip to a size of, if the chip is reduced to a size of 1 inch in any 2 dimensions, that will destroy the EAB overwintering prepupal larvae. Now larger chips, if the EAB is not damaged and they survive, then they can emerge from those larger chips.
larger pieces of bark. They're done feeding, they're done moving. So it's all about whether or not the insect is physically damaged during the chipping process. And so the regulations require they be chipped to a size smaller than 1 inch in any 2 dimensions for it to be non-regulated. If the chips are composted, that may also have an effect if you generate enough heat to kill pre-pupae.
Okay. The next question, Dan, what are your thoughts on planting pure Manchurian or Manchurian crosses in North American forests?
Well, Manchurian ash and the northern treasure and northern gem, both of those are crosses, have been available in the nursery industry for a long time. And they've been planted in arboreta and on street trees. Manchurian ash has been planted in the upper Midwest and Canadian provinces. It's not as desirable as a street tree as the North American ash. But as far as planting in forest, planting an exotic species in forest is it's a value judgment, and so that's something for people to discuss. It's a policy decision, not a scientific decision,
right.
Although it has scientific implications. But I will point out that the hybrid chestnuts are being planted, and the idea with the ash breeding program is to generate a hybrid that has a substantial back crossing so that it's largely a North American genotype with the Asian resistance genes, much like the American chestnut is 15/16 North American.
All right, the next question is from Arkansas. Preservation of genetic diversity is on our minds in Arkansas at the front edge of the invasion. Can you recommend one of the insecticide treatments over others for preserving genetic diversity in mature forests? Well, I think, for various logistical reasons, probably the emamectin benzoate would be the best way to go. It's injected into the tree rather than applied to the soil, which might have some environmental implications, and one treatment is good for 2 to 3 years, and so you can rotate trees that are treated over time, treat a subset of trees each year, and models have shown that you don't have to treat all the trees to get some associational protection of untreated trees. Trunk injections are a little more difficult to make, and there's more timing and weather issues associated with their application. But I think that's the way to go. The emamectin benzoate is also very effective as adults, and they're moving around, and that's going to be the basis of associational protection. As if you're killing adults feeding on a treated tree before they lay their eggs on an untreated tree. All right, a couple things. For those of you that have to get going, it's almost 1. I just put the link for the webinar quiz in the chat box. You can see that. We will keep going. There's several more questions here. Dan, the next one, this is an arborist from a civil engineering firm. How do you properly dispose of EAB-infested trees? Can you burn on the site, or do you have to contact the county, or what's the procedure?
Well, if you're within a quarantine, and you're not moving it outside of a quarantined area, then there is there's no regulatory requirements of how you dispose them. And for example, in Ohio, EAB is so widespread, it doesn't matter how you dispose of them because EAB is already everywhere. If you're going to ship ash outside of a quarantine, then there's certain things that you need to do, like logs. You need to shave an inch off of each side to get rid of the bark and the larvae. Grinding, burning would be effective. I don't know how practical that is in many areas to burn, huge quantities of ash trees. But once EAB becomes established, it's just there, and you just dispose of them as you would any other tree. All right, our next question says, okay, the comment is, as a professional silviculturalist in Connecticut,
I am leaning hard on ash species, particularly on sites where there's good oak, sugar maple, hickory, and pine mixed in. Typically in stands where ash constitutes less than 10% of the basal area. Your thoughts?
I'm not sure I understand that. What was the question in there?
I think they would like to know how you feel about promoting ash species in areas where ash is, already less than 10% of the basal area. And maybe this is a good chance to say, in general, should we be promoting ash or doing silviculture to preserve ash, or are we just at the point where we have to assume it's going to be gone if EAB comes in?
Yeah, well, as far as, unless you treat the trees with insecticide, they'll be killed.
Yeah.
And even in areas where they're a minor component of the forest and at low density, we've, in our sites anyway, EAB found and killed those trees.
Yeah.
So, me personally, if it was less than 10%, I would think the impacts are going to be fairly minimal in a disturbed forest, but it just would depend on what the management objectives are for that forest and what the land, the landowner. And so essentially either the trees are going to die, you can remove them, or you're going to treat them. Those are the only options.
Okay. What role do you see biocontrol agents playing in the continued survival or restoration of ash?
Well, I think that's a really interesting question, and I think how they interact with EAB in those aftermath forests where EAB is at low density and ash has been eliminated except for the saplings is going to be where their greatest potential is, in my opinion. And so, I can't predict. Maybe there'll be some sort of dynamic equilibrium, and maybe not.
Yeah.
But we and others are looking at that now.
Will Manchurian ash naturally hybridize with North American species?
Well, that's a good question, and the research that's going on indicates that it's pretty hard to get them to hybridize. And you can do it. Manchurian and black ash are pretty closely related. They're very closely related phylogenetically, and so they have been able to get them to hybridize. But I don't know if black ash and Manchurian ash have ever existed together in the same environment at numbers, where it would be possible to tell whether they're hybridizing naturally. It'll be interesting to see if that happens in the common garden, but black ash is being eliminated very rapidly from the common gardens, and black ash grows in standing water, and Manchurian ash is, not adapted to that environment. So, but geneticists have hybridized them, so that suggests there may be that potential.
Right. So one other question, and then we have an answer. This question said, are Mississippi, Alabama, South Carolina, and Florida part of the Cooperative Emerald Ash Borer project? The states are blank on the map, if not why. And one of our attendees wrote in the chat window that states become a part of the EAB website, and I assume program, when they have recognized infestations of EAB. Dan, is that how it works?
Yeah, that's a regulatory thing with USDA APHIS, and they work with those state departments of agriculture to survey, and then once EAB is detected in those environments, then it becomes regulated and quarantined, and then a blue line would be drawn around the infestation area. When infestations are small, they only quarantine part of the state. For example, as in Georgia, Arkansas, Louisiana. As EAB continues to spread, as in the case of Ohio, where it's in every county now, the whole state becomes quarantined, so people that, so you don't restrict movement of commerce any more than you have to. So they are monitoring in the states around the infested states, and once it's detected, they'll be added to the program, I'm sure.
Right. Yeah, and Georgia's a good example. We had our infestation around Atlanta, and then we've got the infestation coming down from the north, and right now those two quarantine areas are separated by just a couple county swap, and I would assume within the next year or so, it's maybe 2 years, it's probably going to connect into one big quarantine area. So kind of how it works. Dan, another question. Compared to previous or other forest pests, is EAB cooperation, funding, government or public funds or public outreach any better or different?
Well, boy, I don't really have the data about funding. I mean, EAB funding was good for a while, but as all programs, it's gone down now to pretty low levels, supporting research and outreach compared to what it was, 5 to 10 years ago. There have been other major programs, gypsy moth, for example, in the 70s, southern pine beetle in the 70s and 80s, and mountain pine beetle. So, there have been programs that have been well funded. EAB was, at its peak, I think, received about $25 million a year to support regulatory, mostly to support regulatory, and a few $1 million a year to support research. So it wasn't one of the biggest, but that funding has gone down quite a bit.
Yeah, and I can speak to the public outreach. I think, we've gotten a lot better recently with our public outreach, and we're actually getting ready to conduct an assessment of the Don't Move Firewood campaign. There's big assessments conducted in 2007 and 2010 across several states, and we're getting ready to do a follow-up one across 9 states in the southeast. And so I think once we get that done, we'll have a lot better idea whether or not some of our outreach efforts have really worked.
Good.
So, well, I don't see any more questions coming in. Herbivory question above. Let's see, did I miss a herbivory question? Oh, I'm sorry. Dan, are the impacts of herbivory being monitored or managed in locations dependent upon the orphaned ash cohort for future ash in the forest?
So I'm guessing that question relates to the native herbivores on ash, and I don't know if anybody is looking at native herbivory on ash or not.
All right. Oh, this says primarily white-tailed deer impacts regeneration.
Oh, we don't see a lot of impact of deer on ash regeneration. It doesn't seem, in our area, it doesn't seem to be impacted the way oak is.
Okay.
For example.
So if we can treat a limited number of forest ash trees, which trees would you suggest targeting? The healthiest or the intermediate or some other ones?
Well, I would probably suggest targeting the healthiest, because those would be the ones most likely to reproduce the best.
Yeah, and that's really the big issue, is once these trees die, there's no seed bank. So trying to get those trees that can reproduce to reproduce would be key.
And you have to, make sure that you're treating male and female trees, within a cluster.
Yeah, good point. Good point. What are your thoughts on biocontrol in urban settings, Dan?
Well, I don't see a lot of potential. The biocontrol agents are not having any impact on tree mortality or slowing the spread of EAB, so I don't think they're going to protect trees in an urban environment. I think that if the biocontrol is going to have impact, it's going to be on regeneration in the forest.
With that, I think we'll end this webinar. I want to let everyone know that these slides are available on that forestrywebinars.net webpage. There's a PDF file I added called Emerald Ash Borer Slides. You can get those. You can find, if you want to contact Dr. Herms, you can get his contact information there. And as he mentioned, he'd be happy to talk to you. So, Dan, thanks again. We appreciate everything.
Thank you, and thank everybody. Appreciate